Hello, today's topic is about affinity chromatography and it's lecture 3 related to affinity chromatography. Now, we have been talking about what the principle of affinity chromatography is, what are the terms like ligand and the analyte and the matrix, how to prepare the matrix, then what are the different types of ligands used. So, with respect to purification of biomolecules, the selection and attachment of ligands, it is to be talked about. So when we talk about a ligand, a basic and prior knowledge of the biological specificity of the compound to be purified is a must. In practice, it is frequently possible to select a ligand that displays, say, absolute specificity, that it will bind very selectively and very exclusively to the particular compound. So this selectivity and specific binding is very important. Uh, but at the same time, it is to be kept in mind that when you are going to use it for affinity chromatography purpose and purification of certain biomolecules, then once the column is run, your analyte has anchored or bound to the ligand on the column matrix. The story doesn't stop here. You have to elute the analyte of your interest, and if the interaction is very strong between the analyte and the ligand, then very harsh elution conditions would be required to weaken the interactions between these two components of a biological specificity pair, right? So if the reaction interaction is strong, then strong and harsh treatment is to be given to weaken the interaction. Um, of course, it is possible to select a ligand that has a group selectivity also, right? So it will bind to a closely related group molecules or compounds which have similar structure, or you can say chemical specificity, right? So for example, to say, if your ligand is 5 prime AMP, that is adenine monophosphate 5 AMP. Now, if you take it as a ligand and talk in terms of the molecules which can bind with it, it can reversibly bind to many mm, enzymes which are NAD dependent. So NAD dependent dehydrogenases can selectively bind with 5 prime AMP. It is just because the structurally similar component, all the dehydrogenases they have, NAD dependent dehydrogenases they have, right? So NAD is a common molecule which is having structural similarity among all those NAD dependent dehydrogenases. So it is essential that the ligand should, like, if 5 prime AMP you say, and you try to purify the enzyme, it can bind selectively to any of the members of the uh, uh, type or group of enzyme that is NAD dependent dehydrogenase, right? So when we say this, the concern is you have to have selectivity with reference to the analyte to be purified. So it is essential that the ligand possesses or has a suitable chemical group, a functional group, a group of interaction sites that will not be involved in reversible binding of the ligand. It has to be irreversible. No, not at all. It should be reversible binding. And once the analyte is attached to the ligand, it should be extractable. At the same time, it should have a group of binding sites with which it will anchor onto the matrix also. So the most common such groups which are talked are like, say, NH2 amine or carboxyl group, SH group, phenolic and alcoholic groups, OH groups, right? They are involved in the process. Now, when you talk about attachment of the ligand to the matrix, you should take care that it doesn't prevent binding of the ligand. So when you are having a ligand binding onto the support, it will bind through the interactive forces. And while getting anchored onto the support matrix, the ligand should not compromise on functional sites which would help its binding with the analyte of interest. Right? And this might happen if the ligand is directly implanted or allowed to attach onto the surface of the support matrix. So in such cases, what we can do is we can add the spacer arms, right? So the spacer arm generally is about 6 to 10 carbon atoms, and it has got functional groups on either of its ends. One attaches with the support matrix and one attaches with the analyte under study. So, such what are the spacer arms? How do they function? All this we have discussed in previous lecture. So I'm not going to repeat it to the more details, but this is what we have been talking about when it comes to selection of a ligand and its attachment to the support matrix, and then its attachment to the analyte of your interest, right? So we have taken some examples wherein biological specificity is known and is explored in affinity chromatography. For example, we have talked about antibodies, which are like selectively used in advanced techniques like ELISA. Also, you have different types of ELISA where you are using the specificity of antibodies with the antigen, and in the detection or quantification of antigen you are using it. Similarly, you can use antigen antibody interaction in affinity chromatography. Similarly, you have lectin affinity chromatography, biotin avidin affinity. So all these examples we have to learn today. To say the one, say lectin affinity chromatography. So lectins are, as you know, the proteins which are obtained from plant, animals, and some of the slime modes they produce it. So they have the ability to selectively bind to carbohydrates, and hence they can bind with the glycoproteins also. So proteins with carbohydrate group. So lectins are used to purify and separate the glycoproteins and carbohydrates, right? So the subunits of this material, of course, they can recognize the saccharide component, and not single one, but three different saccharide components can be detected using a lectin, right? And generally, these are say about 40,000 to 4 lakh molecular mass it has. So big molecules, lectins are bigger molecules with molecular mass about 40,000 to 4 lakh daltons. And their ability to recognize and bind specifically to saccharides is very much important and utilized in affinity chromatography for the purification of glycoproteins. And when it comes to glycoproteins, particularly, you can take the example of the receptor proteins which are present on the membrane. So, like that, there are many examples that we have to learn in details today. In the slide that you are seeing, you have another example, that is biotin avidin. What is biotin? We know that it is one of the vitamins which is called as vitamin B7, or also known as vitamin H, right? So this is produced by the cells, and it is known to be a very important cofactor in many of the biochemical enzymatic reactions in the body. Now, biotin and avidin, these are the these are interacting with biological specificity. So, if you see in the schematic representation for it, you will see in the first the biotin molecule, right? So, biotin molecule is attached to the antibody, so it is said to be as conjugated, right? And when you add avidin to that, these biotin molecule get attached to the avidin. So what you see, there are four interaction sites, and at each interaction site you get addition of the complementary molecule, right? So what you are going to have, avidin biotin interaction, and such complexes are going to get formed. When you talk about affinity chromatography, here the affinity tag that we talk about is avidin, right? So tag is the one which is going to be used for mm, uh, uh, purification of the analyte, right? So the tag is generally avidine or streptavidin. Now, streptavidin, as the name indicates, it is obtained from the microorganism, right? So, Streptomyces avidini. So, streptavidin is obtained from this organism. Now, avidin is a molecule which is bigger in size, that is 66 kilodalton, as well as streptavidin, which is 60 kilodalton molecule. What is the difference? The glycan groups are there in avidin, so it is comparatively bigger molecule. Now, when you are using this as a tag, the important thing is, as I said in the beginning of the lecture, that the ligand and analyte should interact, but that interaction should be strong enough to anchor the analyte onto the ligand within the column, but at the same time it should not be very strong that it will be difficult to elute it out. So harsh conditions are to be applied. So when you have to have a purification of biomolecule, the elution condition should be very simple, easy, and non-harsh, right? So conducive environment you have to provide. So when you add a mm, um, biotin as an affinity tag, right? So can we elute your molecule of interest under non harsh conditions? That is very important. So when I'm selecting a pair of any biologically interacting molecules, the molecules with biological specificity, I have to first see that are they interacting, are they strongly interacting, and even if they are strongly interacting, selectively interacting, are they getting departed, or the interaction weakens easily under simple or minimum pH, minimum ionic strength. So it should not hinder or interfere or destroy or disintegrate or denature the biomolecule. That is very important, right? So considering that fact, we have biotin avidin as a good system, and as you know, it has been explored in many of the methods, right from purification to quantification of biomolecules. So when we talk about biotin, the advantage is that it is used in affinity tag, so it has minimal effect on the activity of large molecule, and it can be applied to purify very large biomolecules of the say size 244 dalton. So the proteins can be easily purified, right? So for that purpose, what you need to do is you have to biotinylate the molecule. Matlab, the molecule of your interest should be biotinylated. That means you have to add biotin to that molecule, should that means a complex of analyte and biotin. So how it can be obtained? So maybe you are adding a gene sequence that will allow synthesis of biotinylated molecule, or maybe at post transcriptional level you are going to have addition of certain functional groups, certain sugars, certain carbohydrates, or you can say certain glycoproteins, for example. Similarly, certain other molecules can be conjugated with the protein to make it ready for function. At that stage also biotin can be inserted. So biotinylation is going to happen at gene level or at post translational modification within the cell. So that needs to be incorporated into the cell, how that we will see in the next slide. But biotinylation of the analyte is required. So then what you are going to have, the column is going to be the support matrix, maybe silica gel or agarose, dextran, whatever different matrices we talked about in previous lectures are applicable here also. So to the support matrix, you are going to add the ligand. The ligand is going to be either avidin or streptavidin, right? So your column is going to have avidin or streptavidin activated support matrix, and your analyte is going to have biotinylated molecule. So biotin is the component which will interact with avidin, form a complex. So the protein molecule or a biomolecule which is biotinylated will also get anchored onto this complex, right? Avidin also can be very easily immobilized, via its carbohydrate residues. As I said in the beginning, avidin and streptavidin, they are similar molecules only except there are certain glycans or sugar residues present on avidin, right? So in the column you have avidin activated support matrix, or you have streptavidin activated support matrix. And what is the ligand? Ligand is avidin or streptavidin. What is going to bind is the analyte which is having biotin with it. So it is called as biotinylated biomolecule. Now let's see in the second slide how we are going to really work it out. For example, here you can see the general structure of a plasmid given, right? So this is the original plasmid. The name is uh, P, uh, BR AVM, right? Of course, the name is not really very important. It is one of the case studies that I am sharing with you. But this is PBIR ACM CAM. That is CAM is the name of the antibiotic, which is the marker antibiotic attached to it. So this is this plasmid. To this plasmid, you are going to add this particular tag. Now, in this, this particular tag which you are adding is of a sequence which you are interested to produce, the biomolecule which you want to have, and the tag is that of biotin. So this is gene of your interest that is with the that is added, and the sequence for biotin also should be there, or biotin should be added after this gene is expressed and the product is formed, and post transcriptional post translational modification you should have addition of biotin. So the gene sequence of your interest is added in the plasmid. Now this plasmid is referred to as what it is called as now. It is FEN1 plasmid with kanamycin marker. So it is just you can say plasmid 1 and plasmid 2. No need to remember the names. It is just to uh, share with you a research paper, the findings of the research paper. So the specific names are included here as they have used these particular plasmids in their studies. But you can just say in plasmid first, and it is the modified plasmid. The sequence is added. Now this particular plasmid is written to E. coli and the expression is allowed. So when expression occurs, you are going to have this biotin added to the protein product which is produced by this particular sequence, right? So biotinylation of this particular sequence, that is histidine sumo FEN1, that is what is the name of it. So you need not remember the particular tag or name of the particular plasmid, but you have to remember biotin is added. Now you have to go for a procedure to purify this particular protein. So you will have to lyse the cell and purify the proteins. So for precipitation of the protein will be done first, and then you have to go for purification. So there are going to be hundreds of different proteins which are there in the cell lysate, and you have to purify this particular biotinylated protein. So you have an option that you can, of course, not in a single step. You have to go on purifying proteins with series of chromatographies, but final purification very well maybe certain times in single step can be achieved when you are using affinity chromatography. So in this case, for purification of FN1, you are going to FN1, that protein I'm talking about, you are going to use a chromatography column, which is affinity chromatography column, in which you have the avidin groups which are attached. So as you can see in this schematic diagram, this is beads, right? The gel matrix, support matrix, and to this avidin is hooked this purple color four-armed molecule. In the previous slide, we have seen that what is the structure of avidin molecule. It has got four facets. So that's why the schematically it is shown as a molecule with four facets. So it is anchored onto the gel bead, and now it has got specificity for biotin. So whenever all the proteins samples, what you have extracted from this E. coli will be loaded into this column, and the column will be allowed to run. So as the column runs down, proteins which are having this biotin tag, they will interact with avidin and will get returned onto the column. All other proteins will run down the column. Only your protein of interest, which has this biotin as a tag, or you can say biotinylated protein, will remain in the column, and then you have to elute this column. So Whether the protein is properly purified or not, that can be assessed at every step by electrophoresis, right? So this is the electrophoresis gel, the gel doc that is shared with you. So there are different lanes. So this is your kilodalton. So this is the ladder, first one, right? And then M1, 2, 3, 4, 5, 6, 7, 8 lanes you are seeing. So in the first lane, what you have is soluble fraction of the lysate. So when you kill the lyse the cell, the soluble fraction in the solvent is loaded. So many protein bands have appeared, right? In the second, you have the flow through from the histidine trap affinity column. So different types of affinity columns can be there. So what is histidine trap? That we will also learn. But again, the word is histidine is a tag which we have learned in the previous lectures. Ki what are the different tags that are used? So histidine tag, if that is there, and affinity chromatography only you are running, but see the number of proteins. So your protein of your interest is not very highly purified, right? You are seeing bands of so many of other proteins. So then you go with lane number 3, where you have used this avidine tag, avidine histidine sumo F1 tag. Now in this case, yes, this particular protein is highest in quantity that is purified, and all other bands have faded out. Matlab they were unwanted molecules and they run down the column. Only what remained on the column was this big molecule that is of your interest. You have purified, and a few fine, um, very weak bands of other interfering or related molecules also are seen. So you need to concentrate on this particular fraction and proceed for its elution, right? Of course, can you change the support matrix that is used? That is another thing. Will it lead to further purification? Yes. So here, in place of the support matrix, agarose is used, but not avidin alone, but neutroavidin, which is another derivative of it. It is synthetic, that is attached. And what you see in the lane number 5, all other small bands which we have seen in the previous lane, they also have disappeared. So only single band is seen here. That is how you can select the appropriate tag and appropriate ligand for affinity chromatography, right? Now next we go to some other examples. Now GST tag, that is what we just mentioned in the previous one. So GST is about glutathione S transferase, GST. So this is an enzyme, you know, it is 26 kilodalton protein with 211 amino acids in its polypeptide. And of course, mitochondria and cells, cytosol of eukaryotes and both prokaryotes also you find this particular enzyme. Now, this enzyme has various sources. Natural sources, fine, but recombinant sources also are designed. Now, if you want to purify this particular enzyme from natural source or recombinant organism, how will you proceed? Again, affinity chromatography is used. Now, in this case, glutathione is one of its inherent component, right? So, this glutathione moiety can be reduced. So when it is reduced, it has acid group, sulfahydryl group, right? And that is what is used as the ligand for purification. So separation of glutathione is transferase can be done by GST, as is used as a tag, right? Not only it is about purification of GST proteins, but with GST as a tag, you can purify other biomolecules and other bigger proteins also. So purification of GST, okay, fine. But purification of protein in which GST is a tag, like in previous example, biotin was a tag. Now here it is GST as a tag. So GST you are going to fuse to the protein of your interest. Of course, again, as earlier case during recombination experiments, you can insert the sequence into that, or post-translational modifications. You can see to it that this GST gets attached to the protein of your interest. And then you are going to go for separation. Now look at the diagram. So this is the schematic cross section, you can say, schematic diagram to show the cross section of the column, what is happening in the affinity column. So these rectangular blue shaped structures are nothing but the section of the matrix, right? Support matrix onto which your ligand is attached. Now ligand is what? You have immobilized glutathione. Okay, now this glutathione is attached to this, and when you pour the protein preparation obtained from the cell lysate on the column, it will run down the column, but that time the GST tagged proteins will get anchored onto this your ligand, right? You are seeing a red colored glutathione, that is ligand, GST, that is in green color, and the additional protein to which you have tagged GST, that has got anchored onto your support matrix within the column. Now binding has happened. Now you have to wash the column so that all unwanted analytes, all other proteins will wash down the go down the column along with the washing, will be eluted out of the column, and the column has only now GST tag protein. Now you have to elute this from the column. For the purpose, what you will do, you will add glutathione only, right? So you are going to have the one which is immobilized as glutathione, which is in its reduced form, right? So SH form. So it is going to. So here you are going to have competitive interaction. As in enzymology, you must have heard about competitive inhibitor, the structurally similar molecule with little variation. So here also competitive interaction is taking place. GST tag protein is here, but the GST component is bound to this ligand glutathione. But when you are adding glutathione, which is non-reduced form, it is going to compete with this interaction. It is going to pull this GST towards it and form a complex. So the GST tagged protein of your interest now has got removed from the matrix, and now it will be eluted in the uh, column fraction. It is released, you can go for its analysis. So this was second example of ligand or tag, ligand and tag combination, which is used in affinity chromatography. Here are different examples that I am talking about, the ligands in which are used in purification of proteins. So it is antibody structure, as you see that we have started with. So the amino acid contained in the FC region of the antibody, variable region and constant region, right? Variable arm and constant arm, that is what we talk about. So that in the FC region, when we are talking about, so amino acids which are in the FC region, they have generally the same sequence, whereas the variable arms, as the name suggests, the amino acid sequence is going to be changing, and that gives the selectivity for binding with the antigen, right? So this technique is utilized like in ELISA. Column chromatography also, you can have antibodies explode. So you can see it as immunoaffinity technique, a part of it. Yes. Now further, another system that is used includes dye ligands. Dye ligands. So ligand is a colored molecule, right? So what happened? It was as early as in 1968. Haeckel from Germany, he was working on chromatographic systems, and it was actually gel exclusion chromatography he was working with. But what happened along with the elution fraction, he found a dye molecule, a colored molecule got separated or has got eluted in the fraction. And when he studied, he found that it was blue dextran. So the photograph also is showing the powder of blue dextran, but it is one of the molecular a compound, I should say, compound of interest, which is used in different types of chromatographic support systems, right? So dye ligand, here they were using it in gel chromatography, gel exclusion chromatography. But as they found, along with the protein, that was dye that was bound to it, so there is something happening between this dextran blue dye and the protein. They are interacting, they are forming the bond, and the bond is stable under elution conditions. So this led to the discovery of use of dye ligand chromatography in the affinity chromatography. This affinity between the dextran blue, and that with the protein was explored further. Now, of course, here dye is going to react with protein of your interest. That's fine, but there is the thing is you should be able to remove that dye completely. If it is a natural one, then the molecule may undergo degradation or modifications during the running down of the mobile phase or elution phase, etc. Right? So there are some problems associated with it using as a affinity ligand. So what people have done is they have done some addition to it. What is it? Biomimetic dye ligand. So biomimetic, mimicry आपको पता है, right? So it is not actual but very much same to that. So bio is to that of biological molecules. So biomimetic dye ligand chromatography that was developed. So whatever is the nature of this natural receptor in the molecule, that can be mimicked. And so target protein you have, and it has got a receptor which is going to interact with this dye ligand, right? In this case it is um, dextran blue. So that receptor जो है इसकी interaction is through particular receptor. So you are going to mimic that, right? And a false, or you can say uh, synthesized molecule which is going to have similar analogy with respect to structure can be used as a ligand. So डाई ligand में blue dextran था, लेकिन biomimetic में you can use its derivative which is synthetically prepared. So, do we have columns which use this blue dextran? Yes, the Cibacron Blue 3GA is the commercial name or brand name of a column which uses blue and cephalose. Cephalose is one of the polymers that is used as a support matrix. So, blue cephalose diligand columns are available, and they are commonly used for purification of albumin and different enzymes which are going to be uh, requiring NAD and NADP as their cofactors. Right? Now, third example of the ligand is dyes only. Again, I'm talking about is chlorotriazine polysulfonated aromatic molecules, triazine dyes, triazine dyes, which have been commonly used for purification of albumin, oxidoreductases, or different types of enzymes like lysases, synthetases, and so on. Glycolytic enzymes also can be used. Now, if you see this example of the molecule which is commonly used, here is a chloride ion, right? So this is the di molecule which is going to get anchored onto the support matrix. So during this interaction, a nucleophilic displacement of this chloride ion with the OH group, hydroxyl groups on the support matrix. In previous few lectures, when we talked about TLC specifically, we had talked about silica gel and their SiOH groups, to the details, the pore structure, and the OH groups, how they are present, how they can be modified, all that we have talked. So those OH groups, for example, from silica, SiOH, of course, cellulose also is having it, agarose also is having these OH groups. So the Cl will be displaced due to nucleophilic attack by OH group, and this will get attached to your gel matrix, right? So support जो था उसका जो OH group है उसके साथ ये Cl को misplace करेगा, और ये पूरा molecule will get anchored onto your support matrix. Okay, let's go to the next slide. Can we have some other ligands? Yes, biomolecules also can function like ligands like DNA, RNA, or aptamers. They can be used as ligands for purification of other biomolecules. So in affinity chromatography, you can see here that double-stranded DNA or RNA, that is single-stranded RNA, can be used, and you can use it to purify the proteins which interact with DNA. So DNA binding proteins can be purified. Then DNA repair proteins, of course, they have to interact with DNA. So they also have certain sites which can selectively interact with DNA. So DNA repair proteins and other proteins, say, which interact with DNA in some way or the other, like for ligases, for example, or primases, or maybe those which change its topography, unwind it, helicases. All these can have they have these sites to interact with the DNA, so they can be used. Uh, so this interaction can be used for purification of these enzymes. That means if you want to purify DNA repair proteins from the cell, or primases, helicases, selectively enzymes that interact with DNA, you can take help of DNA, and DNA will be working as a ligand. RNA, DNA also can be used as ligands in the affinity chromatography. Now here, if I talk about single stranded DNA, does it have any use? Of course, as we know, it is going to have the basic structure, one of the two strands you have, so basic structure is same. It is being single stranded, going to self fold, self dimerize at certain places, and transient 3D structures may get produced, as you are seeing in this diagram. So certain loops and certain uh, um, temporary um, double strands, loop and strand structures, they may get formed, right? Now, uh, when we talk about aptamers, they have this capacity to use to be used in affinity chromatography. Why do we need it if DNA is playing its role? Of course, there are certain proteins which cannot have sites for binding with DNA, or no interaction with DNA and RNA. Then what can we do? We can use aptamers. They are nothing but uh, um, the single standard sequence that we are going to use. Now, a potential aptamer sequence is to be identified, right? It can be synthesized in vitro, that is in the laboratory in test tube. By artificial synthesis also you can synthesize it, and then it is used to uh, pair your support matrix. That is, you are going to add it as ligand to the support matrix, right? And then the biomolecule which do not interact with DNA, but this particular sequence, now though it is made up of ATGC, it is single strand, so it has got its certain functional groups open which were not open when it was double strand, and they will interact with your analyte and can be used for purification. So the this lower particular diagram is showing you this is the small helical structure, right? Self uh, dimerizing structure that you are seeing, that is of DNA, which is the aptamer, and this is the biomolecule which interacts with it and gets purified through affinity chromatography. So aptamer affinity chromatography is one of the important variation, you can say, or advancement in affinity chromatography for protein purification, right? Now another one is protein A chromatography. All of us know what is protein A, staphylococci. It produces it, and it is its virulence factor, right? So this particular SPA is going to be, being the virulence factor, is going to interfere with the immune system of the host B cells, and even the antibodies, because it has got specific interaction sites with the antibodies, right? So SPA, that is staphylococcal protein A, you have studied in medical microbiology and immunology, that the structure of antibody and the staphylococcal protein also you are aware about, right? So this staphylococcal protein A is going to bind to the FCY portion of human and animal immunoglobulins. So it is going to interfere with the immune system by binding with the FCY portion of the antibody. Further, it is going to interact with the FAB portion of VH3 of IgM antibodies. So uh, at different locations of the antibodies, the staphylococcal A protein is found to have affinity and binds strongly there and inactivates that antibody. So immune system gets affected, and the bacteria grows, and the disease uh, further occurs. To its vitality is of the patient is threatened in certain cases. Now the thing is, it is going to interact with IgG antibodies also. It is going to interact with IgM antibodies also, and this fragments and subclasses. When we talk about the fragments of antibody and subclasses of antibody, so this staphylococcal protein A is going to interact with the subclasses and fragments of IgG as well. So this is about the medical microbiology part of it, or progression of the disease and virulence factor of the bacteria. But this particular interaction with bacterial protein A, SPA protein of staphylococcus, and that of the antibody, this is biological specificity between these two is explored in chromatography, and that is where it comes as protein A chromatography. Now you are going to use this particular molecule protein A as a part of affinity chromatography system, and that can be used for purification of specific antibodies, right? Monoclonal antibodies, for example, are to be purified. So how are we going to purify the monoclonal antibody? So selective protein which interacts with antibody is to be is to be taken care is to be used in the system, right? So protein A chromatography utilizes protein A as a part of the chromatographic matrix, right? So cell culture, for example, you have, and you have used it for production of certain antibodies. So from cell culture supernatant, you are going to pass down the column, and the antibodies will, which are produced by those cells, they will be anchoring onto your column because of protein A, and you can elute the column at neutral pH. No, no, it cannot. Okay, binding is so strong here that removing this protein A from these bound antibodies becomes sometimes very crucial, and elution requires acidic pH 2.5 to 4, right? So if you talk about protein A resins or protein A columns of affinity chromatography, are they really there? Are they really working? Yes. What are the support matrices used on which it is coated as ligand? So glass and silica based, agarose based, and even organic polymers, as polystyrene based resins, where protein A is used as ligand or attached as ligand are used, and such columns are available on the market. But the concern now here is when you are going to elute the column, the antibody is of your interest, or maybe monoclonal antibodies of your interest, which has lot of therapeutic value and have lot of demand in the market, are to be produced in large quantities. But what about purification? Large quantities can be produced by cell culture systems, animal cell culture systems. But when it comes to purification, when you are using protein A column, it will remove all other impurities. Your antibody will get anchored onto the particular protein A, but how to separate these, how to elute the column, how to get antibody of your interest released from the column, you have to use acidic pH, and in that case it may hamper the functionality, right? So is there any other solution? Of course, protein A, is it not used? Yes, it is used. As I said, there are different types of resins which are prepared using uh, staphylococcal protein A as ligand, right? Now, this protein A, as I said earlier, is used for monoclonal antibody purification. But now, what if the interaction is very strong and the acidic conditions are going to interfere the functionality or structure of the antibody? So advancements are done, which include, say, mixed modes, right? So in mixed modes, what is done is